Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really interesting guest involved in creating a better tomorrow for many patients out there. Today, we have the honor of being joined on the show by Professor Dr. Niels Riedemann, who is Chief Executive Officer and founder of InflaRx, a biopharmaceutical company focused on applying its proprietary anti-complement component 5A and complement component 5A receptor inhibitors to the treatment of various life-threatening or debilitating inflammatory diseases with high unmet medical need. Professor Riedemann has over 15 years of experience in the biotech industry and drug development, as well as 20 years of experience in complement immunology research. He founded InflaRx back in 2007 and has been very instrumental in leading numerous private and public financing rounds of the company. As a physician, Professor Riedemann was appointed as Vice Director of Intensive Care Medicine and led a 50-bed University ICU unit for over 6 years at Friedrich Schiller University. Before that, received his board certification in general surgery upon completing surgical fellowship at the Hannover Medical School, where he also did his Ph.D. And then he spent 3 years doing postdoc work both at the University of Michigan. He also received medical training, Albert Ludwig University, and Stanford. And he has been awarded numerous national and international awards, as well as quite extensive bibliography of peer-reviewed scientific literature. A lot of very important topics to go into today. Honored to have him with us. Professor Dr. Niels Riedemann, thank you so much for taking time to come on the show.
Thank you so much for having me here today, Ira. I'm really excited. Looking forward to speaking about complement a bit.
Absolutely. Before we get into complement, though, I would love to just hand you the floor for a couple of minutes to just learn a little bit more about you in terms of not just what got you into intensive care medicine, but, from the very beginning of all this, you've published quite extensively on the pathobiology of sepsis, which in several of your papers you referred to as this graveyard of pharmaceutical companies. I remember a lot of that from back in the day, but take us, if you would, just into a little bit why you focused on this graveyard from the very beginning of your career.
Yeah, happy to do that. So actually, I wanted to become a general surgeon and a trauma surgeon, and I am board certified general surgeon. As a young surgeon in training back then, as a fellow, I decided to do some break and do some research work. And I joined a great lab at the University of Michigan in Ann Arbor in the US and met the other, founder of the company, Ren Fengu, and I were both postdocs there and doing our postdoc work. And I got fascinated by science. I worked on projects in the lab and was part of the discovery team around the function of C5A and the C5A receptor. It's part of our immune response, our immune system. And I was intrigued how it was involved in creating how your own immune system can create damage to your own organs and tissue. And we worked on different models of sepsis, so basically life-threatening infections where your immune system response gets so furious that it can kill yourself, so to speak. But we also worked on other models of immune complex injury, lung injuries. So we got very interested in the mechanism. And so I got sucked into this meaning of our immune response. And that never left me. So I took this thought back, and Renfeng and I talked about how meaningful the mechanism is and if we can make a drug around it. And I went back to become a surgeon, but then we decided, why don't we bring it to this life-threatening inflammation and try to help patients, and found a company, and we finally did. And I said, well, if we want to be successful where a lot of other companies fail, we need to understand why and if there's a reason. And so I decided to become intensivist to learn the other side of the story, treating these patients on the bedside. And I ran indeed a large academic ICU for over 6 years here in Europe, in Germany. But really, it was also like not just leading it, but I was also involved in clinical trials, in sepsis trials, in ARDS trials, in trials directed, like where you enrolled unconscious, intubated patients. And this is a bit of a different animal when you compare that to other studies. So it has a lot of, I would say, difficulties in managing to get these patients, work around the informed consent with relatives, et cetera, et cetera. So we felt like we figured it out, and that timing of intervention is extremely important, and that wasn't well respected in former trials. So the one thing that we probably underestimated is that by the time we looked for funding and doing larger studies, even though we felt we really figured out a lot of things, so many big companies had failed in the sepsis field that both the big companies weren't investing in that space anymore, but also investors weren't happy with this acute space. So since the mechanism, as I pointed out, is so important for different other areas, we pivoted to devastating immunodermatological indications. And there are some diseases that are very hard to speak their name, but they're really also hard to take as a patient. So high unmet need areas.
Yep.
And then maybe just to close the loop here, long loop, as we speak about it, when COVID hit, right, we had all this knowledge around the work in sepsis. We had tested our anti-C5a antibody in avian flu, septic animals. In this case, it was non-human primates. There was an avian flu strain that broke out in China some 10 years ago. We got into a program there to test the antibody and showed a great reduction of the organ damage done by this virus. So we had a lot of knowledge. And when COVID came, we knew that SARS-CoV-1 showed complement activation. We knew that MERS coronavirus, all in preclinical studies, complement was involved. So we really felt like we have a drug in our hand. We had good ties to Wuhan. We learned during the first days of the pandemic that these patients had complement activation through the roof. I mean, levels of, and we speak about what that means later maybe, but levels of C5a that when you induce them in animals, they will kill the animal.
Yeah.
So when we learned this, we felt, well, we have something in our hands, probably can save lives. And that's why we went back in a way to the sepsis field, if you will, because these are patients that suffer from viral sepsis, obviously.
Right. Right.
So yeah, that's how it all came together. Interesting how life takes you from one end to the other sometimes, but yeah.
Yeah, no, it's impressive in the sense, and I'm glad you stressed that, because when you look at what you're talking about in your publications, you mentioned a lot of work on sepsis, ARDS, which we'll be getting into. I even saw some papers in there on ischemia reperfusion injury, which we don't normally think about connected there, but still a condition where a lot of stuff goes wrong really fast. Inflammation, the immune response, obviously a major part of this problem, as you're mentioning. Take us a little into C5a, because you again, this is something that you've been publishing on for quite a while. There's a really great piece. This is back in 2003, Anti-Inflammatory Strategies for Treatment of Sepsis, when you first start talking about that, and C5a sort of defined as, in its terminology, as an anaphylotoxin. Okay, think anaphylaxis, very bad stuff going wrong as well. Talk about C5a, introduce us to the complement system again, because here's something that, again, we don't talk as much about it, but it is connected to this more ancient innate immune response. So, sort of straddling the border between our immune response and our allergies. Take us into the world of C5a and complement, if you would.
Yeah, absolutely. My pleasure to do that. So when you speak about the immune system, and that's something we often hear in the press, and like, speaking about the immune system without talking about the complement system is like talking about your back without mentioning that there's a spine, right? So the complement system is basically the core of probably the oldest part of the immune system we have, in terms of it's developed already in early species during the development. So it's present, it's inborn. That's why they call it sometimes innate immune system. So it's not the cells, the cellular part. It's something that is in our system, in our cells, in our blood, that can get activated, and that serves a certain purpose. And it's very fast. It's very dangerous if it's overactivating. And it's very non-directed, which is why it can do a lot of good fast, but it can do a lot of bad fast either. So my former boss, Peter Ward at the University of Michigan, Dr. Ward always used to say it's a double-edged sword, right? You kind of want it, but you don't want too much of it. So think of some people have heard about the coagulation system, which it's very similar in principle. Yep. When you cut yourself, there's a system in place in your blood that recognizes through some mediators that you're bleeding. And then there are proteins in your blood that get activated. And the end game is you form a clot and stop the bleeding, right? That's the coagulation system. Now, very similar, the complement system is proteins that are in your blood, in your system, in your tissue, and they can get activated, but not when you cut yourself. More so when there is something in your system, like a foreign surface, like a bacteria, a virus, a parasite, or a protein, an antibody, something that's not yours, that shouldn't be in your system. And that complement system gets activated in a cascaded manner. And the end game is to send a very strong signal to your immune cells to say, there's something in us that shouldn't be there. Get active, get rid of it. So in lay terms, the complement system translates, or is the sensor in our system that tells the immune cells, get active, do something about the foreign stuff in us. Mm-hmm. And we know that can go sideways, because you've heard about autoimmune diseases where your own immune system attacks you. And oftentimes what happens is your immune cells are constantly activated, and they're kind of they have weapons on board to get rid of something that shouldn't be there. But in an autoimmune disease, it should be there. It's just sensed wrongly as foreign. It shouldn't be sensed as foreign. But the immune system gets nevertheless active. And so I call that sometimes dirty bombs, because these immune cells, they have, weapons, like they can release very aggressive enzymes. These are proteins that can, cut other proteins in pieces. They're releasing oxidative radicals that can also destroy all sorts of cells. So you may get rid of the invading microorganism, the virus, or maybe the bacteria, but you may also start damaging your own tissue, because these mechanisms are, like a dirty bomb. Unfortunately, you're hitting a lot of your own guys there, your own cells. Mm-hmm. And so in essence, coming now to C5a, at the very end of that activation process, and probably the best researched part that actually is the biggest booster of the immune response that I know of, those cellular engagement that the cells get active, is C5a. Mm-hmm. And so when you take it out, it's not like your immune system is naked. You still have an immune response. Your cells can still get active, but you don't go into overdrive, right? So in other words, what we're doing is we're taking that booster out to let inflammation take place on a sensible level, but to avoid your system going haywire, starting to damage your own tissue to the point where you develop organ dysfunction and failure. That's the idea. I pause here. There was a lot of information, but I tried to make it more digestible.
It was spot on, and I appreciate that. And again, I think it's important, as you were, making the back spine analogy, because we should hear and understand more about sort of this innate connection, which we don't hear as much about as we should. But I appreciate that. Okay, so that takes us into vilobelimab, which is your first-in-class monoclonal to target C5a. Now I know, reading a little bit about it, and again, I'm not an antibody expert, but I'll let you take this one. So sort of defined as this chimeric IgG4 kappa antibody, which looks like it targets, it's one of the lighter chains of a traditional monoclonal antibody, and in the development of it, while it attacks C5a, it leaves something called C5b9 intact, which, as you were saying, into the tempering, we don't want to destroy all the complement. We want certain components of complement defense intact. It's important.
Yeah.
Talk a little bit about what you developed here. Obviously, the top line, without going too much into antibody biotech. But
Yeah, no. I'm happy to do that. So, modern antibody therapies, and I should probably clarify that first. They're very good instruments because you can very precisely take one player out of the game by the antibody recognizing just that player, in our case, C5a, taking it out and grabbing it and making it impossible to go to its receptor to activate those cells that we've been talking about. Now, that's why antibodies are pretty well-tolerated medicines across the class, generally speaking, right? They're still foreign proteins, so you could still react to them sometimes, have an immune reaction to them. But generally speaking, antibodies are pretty a pretty well-tolerated class of medicines, and they're very good instruments. That is not to be confused. During COVID pandemic, we had like monoclonals. That was like a standing expression. But what they meant is they produced antibodies directed against a specific variant of the virus. So they didn't take out a protein. They were supposed to they were generated to take out that particular virus. Right. And that this virus is a mean thing because it constantly mutates. So as it develops a new chain, as it develops a new mutant, those monoclonals may not see that mutant anymore. That's why, some of these monoclonals, these are antibodies that were generated against the virus, were only working for a specific variant and not for the next one that looked different. Okay? So these are not to be confused. There's a lot of well-tolerated antibodies in the world approved or authorized for use for different diseases. So monoclonal antibody therapy is really a very good tool, in my personal view, if you want to block a certain player in the blood. So that being said, this gives us a good tool. And you mentioned C5a because we only want to take out that booster I was talking about, to not have that strong boosting of cellular immune responses of inflammation causing damage. Of course, the complement system has different meanings also by, for example, it can directly build holes in bacteria, right? Certain bacteria can be brought to a lysis, meaning they can basically no longer maintain their integrity and they just implode, right? And complement can induce that. And that's a function that you don't want to block, especially not when you're looking at a sepsis patient, right? Because you need it as immune defense. So as you said, you want to have a very targeted tool. So ours is very particularly targeted at that part that is cleaved off of the mother molecule C5, which we call C5a. Now it's just a little a, but it's a big difference in the meaning of what happens in your system when that is cleaved off.
Right.
Okay. Pause here.
No, again, it's I thank you for walking us through the story, because that takes us now to sort of the clinical portfolio specifically for this candidate, and again, you've been doing several things here. One, the sort of the emergency use work that occurred in the EU. Which I'd like to go into that first, and a little of your experience there per the EMA. Then we'll talk a little bit about Pyoderma, Gangrenosum, and BARDA. But talk a little about the history here, what it took for you to ultimately get to this Committee for Medicinal Products and gaining the emergency use authorization for COVID ARDS in combination with steroids.
Yeah, happy to do that. So I should mention that, I already mentioned how we came back to in this case, a viral sepsis story. with our approach, with our anti-C5a antibody. And indeed, we decided back then to bring this to COVID patients. And I should mention that we ran probably the largest of its kind global study in patients that received invasive mechanical ventilation or the lung replacement therapy called ECMO. So these are really the most the severely sick patients where the lung has failed to an extent that they need mechanical ventilation or even full replacement therapy, where you give the oxygen outside the human body to the patient. And so we based on the findings there on the mortality benefit, we actually could show in the trial, which was a 23.9% relative difference in survival, so improvement of survival when you had when you were on this drug. We got an emergency use authorization for these particularly sick patients in the United States from the FDA. So for these patients that require invasive ventilation or even the ECMO. And we recently, about a month and a half ago, got the approval in Europe for a similar label, but that's containing SARS-CoV-2 induced moderate to severe ARDS. Now, I should briefly explain. ARDS means acute respiratory distress syndrome. It means your lung is heavily impacted by the virus, by the immune response to the virus. So that's great because there hasn't been an approval in Europe or elsewhere, to my knowledge, where a drug has a label that contains the term ARDS. It's a certainly first step. We only have shown this early signs of efficacy here in patients with SARS-CoV-2. And I don't want to imply that this can be easily just also taken to other areas. Certainly, it's true that the therapy addresses the immune response to the virus. But that doesn't automatically mean that you can replicate an effect in any virus or in any other disease. You always have to show it through clinical studies and prove it, right? So in Europe, this is called an exceptional circumstance approval. That's not so oftentimes seen. That means that the committee probably believes the benefit outweighs the risk. But normally they would require, just like the US FDA, a second study, which because of the fading pandemic couldn't be conducted. And so it's a I would say very exciting stuff for us. It's now a rare incidence, but we're also thinking about how could we further develop with clinical studies and see whether that immune response is similarly meaningful in other diseases that may come up in the future or that exist already. You think about diseases like influenza, avian flu, etc. These are all viruses that can cause damage to the lung and can cause an ARDS and a viral sepsis. But again, you first have to show that in clinical studies that is actually also applicable. And that's something we're interested in. And you mentioned BARDA. I don't know if you want to speak about that very briefly, but the question is, how do you show that it works in other diseases that are similar in principle as well?
Right. Yeah. Well, let's talk about that, because I've had the opportunity to profile members of BARDA and ASPR. David Boucher was on the show a couple months ago, talking about their work, as well as the Strategic National Stockpile, and the fact that, hey, we need these advanced therapeutics for God forbid whatever comes next, or, as you were just mentioning, things that are around that we still, whether it's a severe influenza, and we never know again what form that's going to take year to year. Talk a little bit about your relationship with BARDA, ASPR, to the extent that you can. Nothing obviously confidential, but a little bit about, just, I know you have some clinical work set up there. You're collaborating. Whatever you can tell us about sort of the relationship and the strategy, that'd be great as well.
Yeah, so happy to do that. So, the US authority BARDA is, put together a program called Just Breathe some years ago. And the idea was exactly what you mentioned. Let's develop, potential future therapeutics that could be applied to patients with very bad, in this case, lung diseases. And the focus of Just Breathe is this ARDS, which I mentioned before, right? Which can be introduced by SARS-CoV-2, so by COVID, but also by other viruses, influenza, RSV viruses, but also bacteria, and also chemical injury, like, when you inhale certain chemicals, etc. So this type of lung injury that can occur is a focus on a study that they are starting right now, which is probably the largest conducted study only inside the US in many centers. And that's in the broader ARDS setting. And we were chosen together with two other drug approaches to be tested in this very large, high quality study that they're conducting. And we feel very privileged. We applied for being in the race to be chosen there because this is a fully government funded program. But I think it's very meaningful because I mentioned that this field of sepsis, in this case ARDS, is really difficult to get traction from big pharmaceutical companies or from investors for reasonable reasons, right? It's hard. Development is difficult. History shows lots of failures, and so on and so forth. So the government has seen that, that's my interpretation here, that this is a high unmet need, that with the government funding, there can be potentially development progress, which serves two purposes. One is that, patients with this terrible disease may have a working drug that can reduce, hopefully, mortality one day. And secondly, that's became clear to us after COVID. It would be great to have a pandemic preparedness approach. Yeah. And here we're not talking about something that you develop as you see a new virus, something that is off the shelf available to, for example, address the immune response, which is present maybe in different viruses, so that you could lower mortality right away while you gain a bit time, so to speak, to develop working vaccines or antivirals that are directed against that specific new hit, that new virus. So I think I've been advocating that, well, you could say it's self-serving because I'm the CEO and founder of a company that develops it, but I've been advocating that independently even when we didn't develop in this, because I always felt that maybe I should mention this. When I was an intensivist, I basically lived through some of those outbreaks, for example, the swine flu. And while we talk about it, like it doesn't sound like anything bad, I can tell you, if you have a 38-year-old mom of 3 kids with a full-blown swine flu ARDS in front of you, and you see the lung literally dissolving on the CT scans, you think differently about how it would be if you had anything to offer. And so this was a driving incentive here. And so I think that angle, by the way, is not really fully understood by the public yet, that when you're thinking about a pandemic, that we shouldn't just think about vaccines or just antivirals we should think about from all angles, how do you really contain it so that you don't run behind and have to see those pictures like we all still have in mind from Italy, where they carried out those bags like on trucks. It was horrible.
Yeah, no, it's an excellent point, and I keep pointing sort of the ancientness of the innate immune response and the fact that it's we look at lower organisms and that's their main immune response, and this thing has been evolving over time, and it's smart, and so we have to have multiple ways to interfere at the same time, and this combinatorial concept you're talking about is spot on. So I'm really glad to hear that. Beyond the respiratory diseases and beyond preparedness, you've gotten very active in other areas of unmet need, and one of them I'd like you to talk a little about is Pyoderma, Gangrenosum, and this is something oddly enough that I've seen a couple careers ago, and we think of the usual sort of skin diseases, the psoriasis and the atopic dermatitis and all that, but there's a lot of really bad ones, and not saying those aren't bad, but and the PD is horrible. I'll just put it that way. Pustules and ulcers and blistering, and talk about this, because obviously if you and you're in phase 3 here, if you have success in this extremely unmet dermatological condition, clearly you were going to win across dermatology. So take us a little into this one, if you would. I think it's important.
Yeah, happy. I mean, to do that, because obviously our key focus right now in the company is really the development in these autoimmune and immunodermatological space, I should say, or like derm. And as you mentioned, there are diseases that are quite horrifying, and PG is one of them. Pyoderma gangrenosum. I'm gonna call it PG because it's a bit easier. But I refer to Pyoderma gangrenosum with PG. So PG is a disease where these patients develop those ulcers. There's also a very rare non-ulcerative form, but these ulcers, like, think of them as craters in your skin.
Yep.
And they can grow because they don't heal. Normally, when you have a wound and you have an infection, you may have a little ulcer, but usually it heals. You give some antibiotics, you could wound care, you put some dressing on it, wound dressing on it, you're gonna take care of it, it heals. This is an ulcer that has an underlying disease in the skin. So it's a different story. This disease disables the ulcer from healing. In fact, it can grow, it can further grow. Usually it starts on the lower extremities, on the legs, but it can be everywhere. I've just recently seen an ulcer of a patient that had a gigantic ulcer on the neck, horrifying. Sometimes it leads to be that extremities, like legs, have to be amputated because it's whole circumferential skin loss on a leg. So it's really a horrible disease. It can be multiple ulcers. Typically, it's at least one large ulcer on the lower extremities, so on the legs. These patients, I've worked and I've talked to a patient advocacy group leader in the US once, and she was with us in a meeting with some authorities, and she was saying, no, you may think of this wrongly. This is not a wound. It's like feeling skinned alive 24/7. That's how she explained how she feels about this disease. Yeah. And so that, it's, as you mentioned, there are some horrifying diseases. This is an interesting disease because there's no therapy approved in the US or in Europe. It's an orphan disease. It's relatively rare, not ultra rare, but pretty rare disease. But when you have it, it's a living nightmare.
Yeah. And
so we're running a phase 3 clinical study with our anti-C5a antibody. This is a pivotal study. So it's, we're going to have in the near future in Q2 an interim look to see whether the study should continue if there is efficacy signals at interim, or if it should stop if there's no efficacy signals. Mm-hmm. Why would we do this interim look? Because no one has ever run a real placebo-controlled study. Because it's considered unethical. And the way we worked it out with the US FDA is by saying we're doing this on a background treatment of corticosteroids that are tapered off, to say, okay, this makes it acceptable for the patients to enter the trial, because if they don't approve at certain time points, they will automatically be called a non-responder and get out, so they don't suffer unnecessarily, right? So it's a straightforward trial, but it has never been done in the disease. All the smaller studies done so far were all open label studies. So we are very excited about this, because if our anti-C5a approach were to show efficacy that could give promise to a new treatment, again, it would still need to be approved through the authorities and all that, of course, but it's really a disease that has a very high unmet need. Yeah. We are also developing our small molecule C5a receptor oral drug that we just introduced into phase 2. So that's development-wise still in the earlier stages, but we're also taking it to other skin diseases. Some of them are in the same realm of PG, which is referred to as neutrophilic dermatosis, meaning neutrophils, the white blood cells, are somewhat involved in the disease. Right. And by the way, they're covered with C5a receptors, so they have all antennas everywhere. If they see C5a, they get activated very easily. So that's That's where we are. And I don't know if that kind of closes a bit the loop on PG, but it's really an unmet need, and we are really looking forward to our development efforts there.
Yeah, and yeah, I was just going to go there next because you do have, obviously a couple candidates in the pipeline that are moving things from the parenteral to the oral formulations and these novel small molecule inhibitors. You know, Obviously you're mentioning, PG, and then some of these other conditions which have very long names, and we'll put a link to the pipeline in the bio of the show. In one of the categories of the pipeline, you mentioned additional indications you're thinking of. And I know this is earlier stage, but you have neurology and nephrology on here, amongst others. Obviously, what you're showing here is that these drugs are extremely good at shutting down extremely powerful immune response. Thoughts about sort of the lower level stuff? I mean, we talk about things like inflammaging on the show, and sort of the lower level inflammation that many of us have that are responsible for a lot of sort of the slow chronic degeneration over time. Thoughts in terms of, is there a place for C5a inhibition in not as serious conditions? And because clearly immune response, inflammation, quite behind a lot of the trillions that we spend in this country and abroad. Just general, I mean, I know the earlier molecules, but just your general thoughts on that, if you would.
Yeah, absolutely. So I mentioned before that the mechanism of C5a and C5a receptor inhibition has been very broadly studied and well understood in preclinical studies.
Right.
The reason why we didn't see much more work in humans yet is because we didn't have good tools. We didn't have a fully working C5a antibody, and that has to do with certain biological reasons, which we didn't know when we started developing, but now we believe we understand better, and also with some shortcomings to address this receptor that is everywhere. But the research is all over the place. There's, I don't know, over 6,000 publications on the role of C5a and C5a in receptor in various different disease conditions. Now, I do want to caveat that doesn't mean you can just take a drug from an authorization or an approval and say it works there. But you have to do all the development work and show it behind it. But you mentioned, like, what about the lower grade inflammation or other type? So let me start with the other type first. You mentioned the old name when C5a was discovered. It was called anaphylatoxin.
Yeah.
And there are two parts in it. One is anaphylactic, meaning like allergic shock type of reactions, and toxin means if you have too much of it, it gets dangerous, right? So that angle that it can cause allergic reactions, we haven't talked about that so much. In a way, allergy and inflammation is also very closely linked.
Yep.
But we're testing it right now for skin disease called chronic spontaneous urticaria.
Okay.
So these are these patients that develop these itching hives. And while this is not comparable to like a disease like Pyoderma, Gangrenosum, it is still something that has a high unmet need and bothers you a lot and can be life impacting dramatically. This is a new area where we're looking into the more allergic spectrum of diseases. That is ongoing research because of the known role that C5a can play in allergic reactions. So be really interesting to see first data, hopefully in summer this year.
Right.
And then you mentioned the lower grade inflammation. That's a very interesting topic. I kept that for the last to answer that last year, because it's something that was on our mind for many years already. Because remember we researched sepsis a lot, and one thing we learned when we did the first studies in pulmonary and abdominal septic patients, safety studies and PK studies and so on and so forth, many years back then, we realized that when you looked late, so we had the blood draws like at, when the patient was already well or better at day 28 or even later, that even though overall complement activation wasn't high anymore, even like normal, C5a levels were still high.
Okay.
And what happens when you have constantly higher C5a levels in animal models, it leads to something that we called in preclinical science back then, we called it immunoparalysis. Meaning if your system sees high C5a levels over time, when it sees it acutely, you cause all this damage that we talked about.
Okay.
But when it sees this long term, then these cells stop responding. They're sitting there not doing anything anymore. And so that could explain why patients that survive something like a sepsis may be prone to more infections, because C5a is constantly up, your system is exhausted, and it stops functioning. Then we have those areas of chronic diseases that have these flares, where C5a kicks in a flare. That is very interesting because we see that now there's new research showing that in long COVID, the patients have these always coming back inflammation cycles. You probably heard about that. So that is an area that would be of principal high interest of research to see whether if you blocked it, whether these patients would benefit from that or not. Again, that's research talking. I'm not suggesting you should try like taking the drug for that at all. I'm really trying to say this is a research area where approaches to block C5a or C5aR could be applied in this lower grade or reoccurring like wave type of inflammation for two reasons. Because if you have long-term elevated C5a, that happens after very severe diseases, it could impact your immune system and cause a constant inflammatory stimulus that has negative impacts. And in these wave types of forms, you may just coop those recurrent kind of crisis, if you will. That's a total hypothesis. It's not proven on any level, so it needs to be proven.
Sure.
But you mentioned it, and I think it's really an excellent question, because this is something that's been on our mind for a long time, even though we have to focus our development. So our focus is not there right now. Yeah.
No, again, I understood that, but I like thinking a little bit over the horizon.
Oh yeah,
when I
that's
see
great.
when I see the potential of some of what companies like yours are doing, and again, going back to the immune system being this, we're still learning, just like we're learning about the human brain. It's such a complex mix of dynamics. So I appreciate your feedback and interest in looking at that as well. So thank you for that. Just while we have you today, what else is coming up that we should know about for 2025, either pertaining to clinical development, financing, talks, conferences, anything else that I missed while we have you today that you want to talk about, please?
Yeah, thanks so much. Obviously, we're a public company, so we would make announcements as they are appropriate. Generally speaking, we are at upcoming conferences, and we announced this. For example, the AAD, American Academy of Dermatology, coming up here in next week in Florida. We're going to be showing quite a bit of work from our work in the immunodermatological space, so that could be of interest. There's several abstracts, and our team is there. So if anyone is interested, there's more to be found there on science. We are running a study that we talked about with a small oral C5a receptor antagonist that will be expecting top-line data in summer. And I mentioned CSU and also chronic spontaneous urticaria as a new area of research for us. And then also another skin disease called hidradenitis suppurativa. It's one of those relatively terrible kind of other diseases, hard to speak the name in the first place. But so if you already suffer by trying to pronounce the name, you're going to be suffering even more if you have that disease, I can tell you. So this is very exciting for us. Obviously, we're still small biotech companies, so we have to apply our financial resources really wisely. But I think most excited we are about the running PG phase 3 study that will not read out here, but we have the interim coming up, as I mentioned, and then the new molecule with first data in summer. So certainly an exciting year for us, and I really appreciate being here on tape with you, because I always like speaking about the science and always appreciate questions that go a bit beyond what one is doing. So where can other development take place? So glad we covered that.
Yeah, absolutely. And with that, I just want to ask one more thing while we have you today, because, you know, clearly your expertise in surgery and in intensive care and immunology and drug development and biotech. I also read that you're one hell of a bike mechanic as well, and as well as a triathlete. And I just got to ask, while I have you today, are you getting doing much racing nowadays or competing? I mean, obviously, you got to relieve stress of drug development and raise my blood pressure for quite a few decades there. But anything cool happening on the triathlon front?
Yeah, no, I haven't really been active doing triathlon. That was when I was in the younger years. The one thing that stayed is a passion for running. I'm still a passionate runner. I really repaired bikes to make my living through my studies as a med school student. So that was, and I love bikes. So that's true. That's part of the history. But fair to say that I no longer compete in triathlon. I do run to reduce stress, and I do regular sports. Yeah.
That's important for every biotech CEO to keep stress low, because you definitely get a lot of it. But no, really awesome story, Niels. I appreciate the area so much, and again the diversity of potential indications that you're capable of addressing here, and just wishing you and the company the best with it as we get into 2025 and over that horizon. Again, for everybody that is going to be listening to this episode of the show across the various podcast networks, or who will be watching on our YouTube channel. Again, you've been spending time with Professor Dr. Niels Riedemann, Chief Executive Officer and founder, InflaRx, doing really amazing things, focused on the treatment of these life-threatening, unmet, debilitating inflammatory diseases. Niels, again, I want to thank you for taking the time out of your schedule to talk to us for a little while. Thank you for everything you're up to at InflaRx. And as we like to say on our show, thanks for helping to create a better tomorrow for millions of patients via what you're doing. Really awesome story.
Thanks so much, Ira, for having us. Much appreciated.
Great having
Thank you.
you.